Welcome everyone, and thank you for joining us today for our webinar titled Employing Acoustic, Tactile and PPI Startle Response Procedures in Rodent Behavioral Research. This is Andy Henton from Inside Scientific, and I will be your host for today's event. Our session is sponsored by San Diego Instruments and will focus on 3 modes of startle reflex testing, showcasing the equipment and methods required for these tests and providing discussion as to how and why researchers may employ these procedures depending on their research objectives. First, we will hear from Jason Adair, senior engineer at San Diego Instruments. Jason has been with San Diego Instruments since 1999, and over this time has worked in hundreds of research labs to assist scientists with startle response and animal behavior experiments. His current focus involves overseeing engineering, customer service, and product training. Today, he will provide an introduction to the SR Lab system by San Diego Instruments and discuss the basic principles of acoustic, tactile, and pre-pulse inhibition procedures. Following, we will be joined by Dr. Charles Vorhees, professor in the Division of Neurology at Cincinnati Children's Research Hospital and co-director of the Animal Behavioral Core at the University of Cincinnati. Dr. Vorhees' research focus involves investigating how early insult from drugs, environmental agents, or genetic mutation trigger changes in brain development. This involves the study of brain structure, neurochemistry, and cognitive function with a keen interest in adverse effects associated with learning and memory in animal model systems. The ultimate goal of this research is to develop treatments for developmental brain disorders that will improve long-term outcomes for children. Today, Dr. Vorhees will share some of this research being conducted in his lab, specifically referencing acoustic and tactile startle response procedures that he's employed to investigate neurotoxicity in both juvenile and adult rats.
So some of the most important items in startle testing are, of course, accurate results, reducing variability, and of course, repeatability. Well, how do we achieve this? Let's look at some of the items, which include software flexibility. That is, the ability to clearly define your startle paradigm. Another key element is minimizing stress for the animals during testing. And of course, to see significance in the data, it is important that there can be high throughput by having the ability to test multiple subjects simultaneously. With that said, I'd like to introduce you to the startle response system from San Diego Instruments, better known as SR Lab. The SR Lab is the world's most widely used system and is referenced in many scientific publications. It's also a very mature product and has been manufactured by San Diego Instruments for more than 35 years. The software flexibility that I mentioned in the previous slide is made possible by the use of trial and session definition that allow the creation of a chronological series of events. To minimize stress to the animals are paired with the appropriate size enclosure that allows them just enough room to turn around freely. As you can see here with my mouse on the screen, the animals are inserted in the tube here, and there's doors on the front and back. Also, the sensor that measures the movement is on the bottom here, so the animal is always positioned directly over the sensor for reliable results. I mentioned throughput of many animals by using multiple cabinets, but there's also another way to increase your throughput, and that is by having multiple animal enclosures. When a test ends, a different clean enclosure can be inserted into the cabinet in just a few seconds. To detect the animal's movement, the system utilizes an accelerometer as opposed to a load cell. Dr. Vorhees will talk more about the accelerometer in just a few minutes in his presentation. So let's take a look at what an SR Lab system actually looks like. The major components are a sound attenuating isolation cabinet, appropriate size animal enclosure, a standardization unit, USB interface, and of course software. Let's take a closer look at the primary components that make up the SR Lab cabinet. There is an adjust dial for normalizing sound amplitude in your lab, This is critical because every lab will have different ambient noise levels. Another item of importance is the adjustment dial here used for sensitivity. So what I mean by sensitivity is we need to make sure that all cabinets are equally sensitive to the force exerted by the animals. We accomplish this by using a standardization unit that exerts a known force onto the enclosure, shown here. So let's take a look at a few startle paradigms. Some examples are acoustic startle, tactile startle, and pre-pulse inhibition. Acoustic startle response, often referred to as ASR, can be defined as to elicit a startle response from a sudden acoustic stimulus. The acoustic stimulus used in SR lab is a wide broadband spectrum to ensure that the animal can hear the stimulus. This is done intentionally to ensure that the stimulus is in fact detected by the animal. It is critical that the acoustic stimulus has a very fast rise time to elicit the startle response from the animal. So the acoustic startle is delivered and the measurement from the animal enclosure is recorded by software. Another technique used in startle testing is tactile startle response, or the use of air puffs in place of an acoustic startle pulse. These air puffs can be used alone or with pre-pulses. With that said, let's look at pre-pulse inhibition. Pre-pulse inhibition is when a weak acoustic stimulus is delivered prior to the startle pulse and inhibits the startle reflex. The pre-pulses are typically very short in duration and range anywhere from 3 decibels above background level to as high as 16 decibels or maybe even a little higher. Startle pulses are typically in the 110 to 120 decibel range. If we were to design a pre-paradigm using the SR lab, this is how we would accomplish it, by constructing trial and session definitions. So the first step would be to build your trial definitions. So if you look at this screen here, basically what we're doing is we're starting at time 0 and placing events that will be delivered to the animal in sequence here by utilizing commands available here on the right. The variability between different users is minimized as the trial definitions are saved off as discrete files, so it is not necessary to build your definitions more than once. Once these trial definitions or building blocks are complete, you can build your session definitions. These session definitions will serve as your templates for future use. So let's look at one of these here. So some of the key fields are number of samples, in other words, how long do you want to record data from the enclosure. Acclimation time, that is, place the animal in the box and let him get comfortable before the first trial begins. Background level, this is used to create an artificial noise floor to mask out any noises from the lab. And then there's other things here like number of runs, how many times do you want to repeat this. And then off to the right here is a trial list. So the trial definitions that you've constructed earlier are now placed here and will run in order as they're inserted here. Another component of the session definition are intertrial intervals. So there's a tool here that allows you to enter ITIs manually, or you can use the number generator tool that's included. Accurate results are best obtained when the exact same method is used to run startle testing. So these session definitions serve as templates, so they lay the groundwork in exactly the same way regardless of the animal group or person conducting the experiment. So essentially, once you've saved this session definition here, it gets used over and over regardless of the user. To verify that the equipment is calibrated properly, we have a thorough diagnostics application tool that is used. To minimize system variability, the diagnostics application is used to check decibel levels and also baseline values of the SR lab cabinets. The SR Lab is a very sensitive piece of equipment designed to detect the smallest of movements, so it's critical to make sure that the platform holding the candidate is stable. So we've looked at several things, including experimental design and calibration, but now let's look at some data. The data that you see when running the experiment real-time is the same waveform you will see in the data. This is very important to the user because you're looking at a snapshot of the data real-time, so there will be no surprises later when you review the data. As you can see, the continuous response is recorded, but there are a few parameters that are of most interest. Shown down here, if you can see, start, Vmax, Tmax, and average. So let's look at these values in an exported format. The exported data carries all the header information from the session, as you can see here. But let's focus on vstart, Vmax, Tmax, and average, which are in columns 12 through 15 here. Let me show you what these metrics actually are. So if you look at this particular waveform, vstart is essentially the value that's detected on the enclosure at time 0. In other words, was the animal still when the session began. Vmax will be your max data point detected within that particular record window. Tmax will be the time at which Vmax occurred, and average will be the average of however long your record data window is. Apart from acoustic startle, tactile startle, and pre-pulse inhibition, there are other applications of startle testing, such as fear-potentiated paradigms that utilize electric foot shocks. The duration and intensity of these foot shocks can be designed in the trial definitions. Here is just a quick of an enclosure outfitted with foot shock. As you can see here, here's the rods that deliver the shock to the animal. And like I said, you can set the shock level manually here, or you can set it within software. Dr. Vorhees is going to show us some data from research in his lab, so I just wanted to share a photo of how the air puff is delivered to the animal and what that looks like. So the animal's in the tube here, and the air is delivered right on top here, so the air will be delivered to his back. And keep in mind that the animal will be centered here between these slots where the doors are inserted, which is not shown here. So we have looked at several applications of startle testing with the SR Lab, but there are plenty more. These are just a few of the accessories that SDI offers. So please feel free to download the datasheet available in the handout section of this webinar, off to the right here. This presentation was intended to provide a brief overview demonstrating some of the key elements of startle testing used to obtain good data suitable for publishing. Thanks so much for your time.
Well, it's my pleasure to join you today to talk a little about some data that we have on acoustic startle to show you how we use the system, the SR Lab system. Okay, so without further ado, I'll first start with showing you what startle is. Here's an armadillo showing a classic startle response, and there's a cat showing a startle response. Cats are particularly striking in the kind of startle reflexes that you will see in them at times. But we're going to focus on rodents, and here is a time-lapse photograph of a rat showing an acoustic startle response. As you move down from the first to the subsequent frames, you see that the animal the response starts up at the head and neck. You can see it most clearly here as the neck begins to contract and the forelimbs begin to contract. The body sort of contracts as you get the response of the diaphragm and the rest of the forelimbs. And here's sort of the peak of the response. And then as the response moves, it moves from a rostral to a caudal direction, and ultimately you get the hind limb involvement in the contractions. So that's the phenomenon itself. And I did want to mention briefly that between the two major methods for detecting a startle response, one is the load cell method and the other is the use of an accelerometer. Load cells are basically the same kind of detection device that's used in an analytical balance or a scale, and responds to, sends out electric current in proportion to the weight of the animal on the load cell. So when you have load cells in a startle system, the first thing that happens is part of your dynamic range is taken up by the animal's body weight, and then you have to subtract this value from any change that occurs as a result of the startle reflex. So you can see that in this case, this example, the response goes up and like this. One of the things that's interesting about load cells is they tend to be slower in response, and so you don't get all of the response characteristics that you can get out of an accelerometer. An accelerometer, you put the animal on it and it gives a momentary reading and then it returns to 0, so that it only responds to a change, it only puts out voltage in response to a change in the pressure on the accelerometer. And so you can get a much more detailed picture of the response characteristics. So this peak here, the diaphragm and forelimb contraction, is the approximate equivalent to what you get in a load cell over here with this peak. But you can see you don't get all the same peaks with a load cell. So this is from a review from Gomez Nieto, and I think it's one of the really best reviews of the acoustic startle circuitry that I've seen. So what happens is the acoustic signal first comes to the cochlea, then it is transmitted by the cochlear root neuron. Let me see if there's my pointer, and it crosses over and eventually goes to these giant cells in the PNC. And then from there, it descends through the spinal cord and eventually reaches the neuromuscular junction and then out to the actual muscles, which exhibit the reflex. And what's interesting is, electrophysiologically, the latency to each of these nuclei has been measured rather precisely. So about 2.2 seconds to the cochlear root neuron, about 5.2 seconds to reach the giant cells of the PNC, another 6 to 8 seconds to reach the muscle, and then of course there's another few seconds for the muscle to actually contract. So what I did here is I took that same response curve that you saw previously, and using the review data for the physiology, I sort of put on here for you to see when these different events are happening. So the cochlear root nucleus delay is here, the PNC delay is about here, the neuromuscular junction is about here, and then by the time the muscle contracts, the SR lab picks up the response at about 14 milliseconds and goes from there. From the best that we can tell, the neck contraction tends to be the first wave. The largest wave occurs when the diaphragm and forelimbs contract. Then you have, once those forelimbs contract, then they extend again, and then it moves to the hind limbs. You get hind limb contraction, and this is probably hind limb extension. So this is the system that Jason showed you. This is an older cabinet where the controls are on the side rather than the front. I use it because it shows an animal in there so you can see. And this is sort of a conceptual drawing to show you how the animal is arranged on this platform that has the accelerometer attached to the underside and with a speaker up in the ceiling unit. So I'm going to just show you some examples of how we use the system, and in this particular line of research we've been studying pyrethroids, which are widely used insecticides, and of course as insecticides they're designed to be neurotoxic. Pyrethroids are divided into two classes, type 1s and type 2. Both types affect voltage sensitive sodium channels, but they produce different effects. Type 1s produce repetitive firing, and type 2s produce depolarization, which is quite severe and actually produces a neurotransmission block. Type 1s increase acoustic startle, and type 2s inhibit acoustic startle. There's been a lot of data collected on pyrethroids over the last 50 years, and quite a bit of it in startle. However, very little data has been collected in young animals, and so our research was focused on testing the idea of, are young animals more sensitive, and that would be useful to know if young animals are more sensitive to the extent that they are predictors of exposure to children. This is a significant issue since children actually get exposed to many insecticides more than adults, because the fact that they get down on the ground and they crawl around. And pyrethroids are used in residential use quite extensively, in insect sprays, on lawns, on pets, treatment of head lice. So they're widely used products. So in this research, we used a prototypical type 1 permethrin and a prototypical type 2, which is deltamethrin. We treated them only once by gavage with one of several doses, and then we tested them repeatedly at 2, 4, 6, and 8 hours after treatment. We used adult Sprague-Dawley male rats. As Jason described, you set up a session. We used a 5-minute acclimation period. For the permethrin studies, we used 100 trials. For deltamethrin, 50 trials. I'll explain why a little later we changed the number of trials. We used a fixed intertrial interval in these studies of 20 seconds. Our stimulus was 115 decibels on the sound-specific level on the sound level meter. The recording window was 100 milliseconds. And we, in these studies, found that rather than matching the groups by body weight, because body weight is not particularly, in samples that we were using, was not correlated with startle response, what we did is we matched the animals on the base of a startle pretest. So we actually put them into a session the day before we were testing the compound, just gave them a gavage of corn oil, and then tested them, and then used those data to match and assign animals to groups. So this is what permethrin looks like. As I said before, it increases the acoustic startle response. At very high doses, this compound is associated with what has been described in the literature as a T syndrome, in which it will induce tremors. So here's our data on the adult effects. As you can see, there are characteristics of the study design are provided over here on the left. And at 2 hours after they are exposed, you see no change in the startle response. However, at 4 hours, you can see that the 2 higher dose groups show a significant increase. This continues at 6 hours, and the low dose group starts to show a rise, which becomes significant at 8 hours, and the other 2 groups remain high. Since we didn't know what to expect in young animals, we started by testing 3 different age groups: postnatal day 15, 17, and 21. We tested higher doses. We weren't sure where to start the dosing, but as you can see, and the other issue, of course, is being able to detect the startle response in animals this young. So what we did is we gave higher doses than we had used in the adults, 120, 150, and 180. And you can see that at these ages you get very clear effects, but the effects down here look a little bit smaller. However, we made those were done in those preliminary data were done in somewhat smaller groups in a relatively small number of litters, and in those studies we were just testing pups regardless of litter membership. We weren't taking litter into account in those initial studies. When we got to the final study in the 15-day-old animals, we used 16 litters. We tested 1 male and 1 female from each litter at each dose level. So 32 pups altogether per dose, with sex as a factor in the analysis. So here's what we got. So as you can see that at this age, the increase in the acoustic startle reflex occurs earlier. It's clearly present dose dependently at 2 hours, but something interesting happens at 4 hours. The lowest dose group continues to show an increase, but the two higher dose groups start to show a switch and show a downward trend. This is actually a trend upward. This is a trend downward. At 6 hours, the low dose group continues to show an increase, but now the high dose group shows a downward trend, which becomes significant at 8 hours. So there's a switch in the pattern in young animals from facilitation of startle to inhibition of startle, which we think reflects greater toxicity in the younger animals. So here are the adult data on the left and the P15 data on the right, just so that you can see them side by side to see that they show, in fact, different patterns. Only facilitation in adults and a mixture of facilitation and inhibition in the young animals. And then moving on to deltamethrin, as I said before, that's a type 2. It causes depolarization block. At high doses, it produces what's called a CS syndrome, in which the animals exhibit chorioathetosis, or a writhing movement, and salivation. So here are the adult data. Now, since adult deltamethrin suppresses startle, one of the things we didn't want was to work against ourselves by having habituation cause a decrease over the repetitive time intervals. So we shortened the number of trials to 50 trials per session to sort of reduce the habituation effect. And that worked pretty well. As you can see here, we get fairly modest habituation. It starts out at about 1,200, drops a little below 1,000 here, but hovers around 1,000 at these longer intervals. But you can see we get a nice dose-dependent suppression of startle that's a function of dose. And notice that these doses, this dose is it's not a high dose, but it's much higher than what I'm going to show you here in the juvenile animals. And that group continues to be affected at all of the time intervals, and that's, as does the 8 milligram group. So here's what happens in the postnatal day 15 animals. We had to drop the dose down because the dose that we were using in the adults was in fact too toxic, and the younger animals couldn't tolerate it. So when we drop the doses down to 1, 2, and 4 milligrams per kilogram, you see that the suppression of startle starts immediately at 2 hours and becomes progressively more severe at 4, 6, and 8 hours. So from these data, here they are side by side, we concluded that developing animals are in fact more sensitive to deltamethrin than are adults. So to sort of conclude on that, these are the first data of age-dependent and time-dependent effects of type 1 and type 2 pyrethroids in rats as young as postnatal day 15. And the data clearly show that young rats are several-fold more sensitive than adults. And the nice thing about ASR is it allowed us to do a time course study of the effects. So, as I said, one of the advantages of Startle is its test-retest stability, which is rather unique among behavioral test methods. This isn't true for locomotor activity, which shows marked habituation. If you, of course, put animals in a locomotor test for that period of time across 8 hours, they would basically hardly be moving by the end. Learning tests show, of course, learning, and therefore the response changes as a function of time and experience. Now, schedule-controlled operant behavior, where you establish a stable baseline, would be ideal for a study like this in adult animals, but it's not feasible to do it in 15-day-old animals. Here's the startle circuitry shown again. This is what I showed you before from the review paper. And here it's shown, however, for the sensory input pathway for tactile startle. So the two, of course, the efferent pathway is exactly the same. So here's just some control animals, adult rats, tested in alternate ASR and TSR. What we gave them is 10 ASR trials, then 10 TSR trials, and we repeated this across 5 complete blocks of trials and then averaged those trials for you to see here. What you can see is that the tactile response is somewhere between 2, 2.5, and in some studies as high as 3 times higher amplitude of response than you get out of acoustic. And here it is again in another group of animals, and here we gave actually deltamethrin at 8 milligrams per kilogram in adult animals, and you can see the suppression of the response here in the acoustic. It doesn't really show up very good, but that's partly because the ordinate is compressed to be able to show this. So again, it looks like TSR has some potential. Then we wanted to do it, of course, in young animals, because young animals is where it's harder to detect an acoustic startle response. And I intentionally put these on the same scale so that you could see the relative effect. And here's the lower dose of 2 milligrams per kilogram of deltamethrin in 15-day-old animals, and you can see the suppression here. And then you see it also here, but the baseline is much higher. So I have often gotten the question of what is the effect of the intertrial interval, and this is a little experiment we did in control animals to look at that. We used 50 trial sessions where we tested 3 different intertrial intervals, 8 seconds, and we use that because it's common in the literature, 20 seconds, or we used the SR Lab feature of choosing the so-called random, and those are the particular intervals it uses with an average of 16 seconds, and did an analysis of variance on Vmax output, and as you can see the means are essentially the same, and an ANOVA test showed an F value that was not even close to significant. So within that range, our conclusion is that intertrial interval doesn't make a significant difference. I often get asked about which dependent measure should be used, and the system provides the maximum response amplitude, Vmax, the average response amplitude, as Jason mentioned, and so across a series of experiments what we did was did correlation coefficients on these 2 measures, and what we find is that, at least in our hands, we get correlation coefficients from 0.96 to 0.98, or very close to unity. So basically these 2 measures are providing essentially identical information. I wanted to show you an example of pre-pulse inhibition. This happens to be in mice, and this is an experiment we did with collaborators where we knocked out this gene, NPAS3, which is thought to be associated with schizophrenia, and what you see is the pre-pulses suppressing the acoustic startle response, the unmodified response here, Vmax, and it did the same in the knockout animals, but notice that the knockout animals have a much higher baseline to begin with. And this just gives some of the details. We use a 70 millisecond time window between the onset of the pre-pulse and the onset of the pulse. And here's a similar kind of experiment done in Sprague-Dawley rats, and these animals were exposed prenatally to citalopram, the antidepressant, or saline, and now they're being tested as adults, and you can see the effect in the saline animals here, and notice that the citalopram treated animals show a marked increase in their startle response, and they show a pre-pulse inhibition effect. Now you will see in the literature many people use percent inhibition, and there's certainly nothing wrong with looking at it that way, but I would suggest that if you decide to show percent inhibition, you show their unmodified response, since obviously the suppression here is not the same as it is here, given the different baselines. So it's always important to tell the reader what the baseline is. So these are our conclusions about ASR and TSR. ASR is a very versatile measure. It's been validated across many species, mice, rats, and definitely in humans. You can detect both facilitation or inhibition using it. You can measure habituation. You can use that as your primary outcome measure if you wish. You can use various kinds of reflex modification, the most common being pre-pulse inhibition, and it has a known neural pathway. At least it does for the basic reflex. And when you use pre-pulse inhibition, what you're doing is there are higher centers in the brain, starting with the inferior colliculi, which produce a suppressing effect on the basic reflex. And there actually are thought to be even higher centers in the cortex which can suppress or contribute to the suppression or modification of the acoustic startle response. When it comes to TSR, it's, as I've noted, a stronger response. It shows minimal habituation, which could be an advantage in some experiments. The SR lab will certainly allow you to set up a reflex modification like pre-pulse inhibition with TSR. We haven't explored this yet, but it's something we intend to do. Has a very similar pathway. We think it's promising. There's not been a whole lot of studies published on TSR, so I think it needs more research, but I think it's an area that deserves further attention. And there again is the acoustic startle response, so I thank you for your attention.
First question, can the presenters explain what acoustic startle rise time is and why this is important? And Jason, I'll ask that maybe you lead this one.
Sure. Yeah, that's a great question. That's a critical metric when it comes to eliciting the startle response from the animal. Generally speaking, the rise time is the time it takes for sound to change from essentially a low volume, like background, up to the desired decibel level. Particularly with the SR lab, it has a rise time of about 1.5 milliseconds. So it's happening really quick.
Yeah, and I would just add that frequently reviewers will ask the question, if you report your startle data, they'll ask you what was the rise time of the acoustic signal. And so that's a good piece of information to have at your disposal when you're publishing your data.
Perfect. All right, perfect. Great answer. Next one. We've got a question. How do you calibrate sound levels between boxes? So again, Jason, maybe you can lead this one.
Another good question. So it's very important that the sound being delivered to the animals is uniform across all boxes. So to accomplish this, we utilize a sound level meter from a third-party source and place that inside the SR Lab cabinet. And then in software, we have a range that goes from 0 up to 750 that you kind of dial in so you can land directly on the decibel level that you need. So the sound calibration doesn't need to be performed very often, but it's one of those things that should be checked periodically.
Yes, and typically what we do is we will check that at the beginning of a new experiment. That's the typical time we do it. Or if it's an experiment over a period of many months, then we will check it every few weeks.
Okay, great. All right, so Roland has asked, is a decreased startle response measured in transgenic mice purely based on a misconnection in the startle circuitry system, or could there be other factors at play here? Again, Jason, what are your thoughts on this?
Yeah, that's a good question. Might need a little more details from Roland on what he's referencing about the misconnect. But one of the things that we see and get questions on quite often is about, something to look at is the hearing affected in the mouse. We see in some strains of mice like C57 that as they age, their hearing decreases significantly. So that's something that we could talk with Roland about and dig a little deeper into.
Okay, yeah, I mean we've seen that in C57 mice. I actually had it on the slide, but I didn't highlight it as I spoke. But we definitely see that as C57 mice get older, they show hearing loss, which will interfere with your ability to do acoustic startle. And one of the things that I would also say about mice in general is that when you're testing mice, look through your data for non-responders, and that is often a hint that you have hearing loss in some of your animals.
Okay, great. That's great feedback. Another question. So David has asked, this is a question for you, Dr. Voorhees. Were the same subjects used for each time point when testing PRM and DLM?
Oh, and at 2, 4, 6, and 8 hours, yes.
Yes.
Those were the same animals. So that's one of the things that was a strength of the Startle is that you can test the reflex over and over and over again with very little loss of response amplitude. I mean, you get a little bit of decline due to habituation, but with Startle, it's quite minimal.
Perfect. And actually, this plays into your answer here. We've got a question that's come in from Mark, and he would like to know, in your experience, how does the Startle response correlate with other behavioral measures? And he's referenced here an elevated plus maze, but I think what he's really looking to get is your opinion on using Startle as an additional component, or an additional behavioral test in a battery for animals undergoing stress. Stress is his particular focus. Can you share your comments on that?
Well, as a general rule, stress will increase the Startle response, at least acute stress will. Chronic stress may increase Startle also, but it will depend to some extent on how long after you stop the stressor and when you test the Startle response. But Startle is quite different than anxiety measures like elevated plus or elevated zero maze. And so it's really quite a different measure. And so I would say that you're really measuring two different things. One is measuring anxiety, and the other is a reflex which can be influenced by stress, but is not considered a direct measure of anxiety.
Okay. Very good feedback. Jason, anything to add there?
Nothing to add on that one.
Okay. Sounds good. Moving on to our next question. Esteban would like to know what type of stimulation frequencies and perhaps procedures can be achieved with the air puff system. And Jason, maybe you could talk about this, and Chip, maybe chime in on maybe what you've done in your lab as well.
So he's curious about the kind of the frequency duration of the air puff that's delivered to the animal?
Yeah, he has specifically said stimulation frequencies. I thought that it might also be interpreted as the procedures that might be planned, like repetitive frequency of a puff.
Oh, okay.
If that makes sense. But
Chip may know more than myself about this one, but the delivery of the air puffs are typically very short in duration.
Okay.
But as far as the frequency that are used, Chip can probably speak to that a little better.
Well, we typically use 20 milliseconds for the air puff. I mean, 20 to 40 milliseconds is the typical range. One thing I would point out about tactile startle is that because the air pressure has to be controlled by a solenoid, there is in fact an acoustic signal that comes off of the solenoid. It actually clicks open and clicks shut very rapidly. And it's a mechanical device, so it makes noise. And so what we do with Tactile is we also run trials where we basically measure just the effect of the solenoid clicking without any air. And then we do that just so that we can understand whether the response is in fact influenced by the sound that the clicking solenoid makes. And at least in our hands, it's not much of an effect, but it's something you should look at in your own system if you're going to do air puff startle. And Tactile, the range of air pressures, the PSI that you use can go anywhere from, I think on Jason's slide it was like 60 PSI. I think we're currently using 80. It doesn't really matter once you get up above about 50 or 60. It'll elicit a strong response. You can go higher and it just doesn't make much additional difference.
okay, that's great feedback. Thanks for that. We've got a question here, a few questions, one from Heidi. She's just asking about the body weight of young mice. So, she's heard that researchers have experienced challenges when running mice younger than 45 days due to weight. Is there a prescribed lower limit for weight and or age? So again, Jason, is there kind of a standard response or experience from the SDI side of maybe Dr. Vorhees, you could comment on maybe what you've experienced in the lab as far as weight and age?
There is definitely a range. On the lower end of things, the mice, if body weight's around 10 grams and less are a little tricky to detect. Anything above 10 grams, the researcher would want to use something like our small enclosures, which is adjusted for that particular body weight to pick up those very small movements from those guys. So
Yeah.
kind of 10 grams and above is kind of a rule we go by.
Okay.
Yeah. Now we haven't done a lot of testing in young mice. Most of the time when we test mice, they're adolescent or adult. Now in rats we do test young rats, but yeah, young mice present a challenge. There are a couple things that you can do. First of all, you'd want to get the smallest holder or enclosure from SDI. They do make one that is a small one intended for small and young animals. But the other thing we do is we adjust the sensitivity of the system. So in an adult, we generally set the sensitivity either at SDI's recommended value of 200, or for some experiments where we know that the compound is going to suppress the response, we've shifted that upward to 300. In young animals, however, we set it even higher at 700. So, and that really helps in the detection sensitivity of the apparatus.
Okay, perfect. Great, thanks for that that answer. Just looking through the list of questions that are coming in, again, folks, thank you for everybody that's contributed and keep them coming. We've got another question from Heidi. I'm curious here whether she's referencing ITI or ISI. She's written ISI, so can someone comment about ISI and PPI with mice? So pre-pulse inhibition with mice. But is it perhaps ITI? Is there a short form of ISI?
Well, ISI, the interstimulus interval is
Okay.
we typically use 70 milliseconds, but there's a broad range that you can use. People use anywhere from about 50 milliseconds. I've seen people use 100 milliseconds or even higher.
Okay.
What you want to do is test it out in your own hands and see what works best. 70 milliseconds we find works well. 100 milliseconds also works quite well, and that's measured from pre-pulse onset to startle signal onset. So it's onset to onset is that number of seconds. So if the onset is at time 0 and it's on for 20 milliseconds, then you'll have 50 milliseconds of no sound, and then the startle stimulus comes on. So it's onset to onset being 70 milliseconds.
Okay.
And we see that quite a bit. Chip is using 70 milliseconds for the ISI. From other labs, I've seen anything between 60 and 120 is a very common ISI.
Okay. Very good. Thanks for the feedback there. Maybe switching gears a bit, just going back to the SR Lab system. Alicia has asked, as a current customer, it appears that to her at least there's a newer version of perhaps the software. Does that make sense? And if so, how can current users access the latest version of the software? Are there any additional features that may be available?
Okay. So if Alicia has the system or has had it for a little while, you basically get a newer version within 1 year if we released it. So I'll have to look up and see if there's any feature change of what she has as opposed to what's out now.
Okay.
We rolled out a new version a few months ago. Yeah, so I'll have to check to see if there's a feature, increase or not.
Sure. Yeah, it sounds like if there's a variety of different versions, and best to check this based on what a current user is working with now and what the latest one offers. Very
Yeah, and
good.
a lot of that is operating system compatibility, with, always playing catch up with Microsoft to keep up with their operating systems.
That's
So,
true. That's true. Very good. Okay, so definitely a direct follow-up with you, Alicia, and make sure that Jason connects with you after the webinar. Again, just looking through the questions here, guys. Give me a moment. Okay, so another question from Roland, actually. He has asked if ASR responses are very low, which they might be in young mice, is enhanced sensitivity calibration a better option to use over switching to TSR? So maybe, Chip, what's your opinion on this?
Well, yeah, I would, if he's getting very low responses, I would certainly try increasing the sensitivity setting. But the other advantage of running a session on his mice with TSR would be it would help him determine whether a hearing deficit is playing a part in his acoustic startle response. So, if TSR and you're getting good responses and you run ASR and you're getting very weak responses, I would suspect that maybe some hearing loss is occurring.
Okay. That's great recommendation. Anything additional from you, Jason, on that one?
no, I think increasing the sensitivity, there should be some room for him to try that easily.
Okay.
So that's something that we could definitely help him with, to increase sensitivity and see if that helps with his ASR sessions.
Okay, perfect. And also just a general question again about system setup and also operating capacity. For high throughput studies, is there a maximum amount of boxes that can be put together and tested simultaneously with all the data coming into like one software at a time? So are there any limits there, or?
So we limit the system to 16, which is quite a few. So we can handle 16 through the USB interface box that I showed on one of the slides, and so that allows you to use a laptop or a desktop, of course.
Okay.
But when you approach 16, I think you're getting to more of an issue with lab space than you are with
Sure.
capacity on our end. But yeah, we settled at 16 cabinets total.
And so, and that's into one operating system as well, one computer connection, one.
That's right.
Okay.
Yeah,
so in theory, if someone wants to go
right.
above that, an additional operating system as a standalone is possible. Okay.
You got it. Yeah.
Great. Great. Okay. Well, just one final question from Ivana. The question is, would TSR be better than ASR when testing fear-related memory after trauma or stress in rats? So what's your thoughts on this, Chip?
Well, I don't have any experience in that particular context, so
Okay.
I'm not really sure how to advise the questioner on that point. But I think it would be a situation where you could try them both. Now, obviously, there's some additional cost and complexity in setting up TSR. How you set up TSR, you need a pressure line, an air pressure line. And depending on what kind of lab you're in, that could be either quite easy or a little more challenging.
Right.
If you have to buy compressed air tanks and bring them into the lab and getting all the tubing set up, it is a little more complicated. We happen to be in a setting where we're connected to a hospital, so medical air is available, and all we had to do was run a line to our air distribution system and then connect it through the tubing system that San Diego Instruments provides. So I think it depends a little bit on how much you want to explore this area, but I would think it would, depending on how it works with the ASR. I'd try it with ASR first if you're set up for it. That's a lot easier.
Mm-hmm.
But if you're not getting the kind of results you are expecting or predicting, I would then consider trying it with TSR.
Great. It's Good recommendation. And actually, we're gonna do one more question. Actually, a good question's come in from Sean. So Sean has asked, during the acclimation, is it typical to generate background noise, and how critical is this? So maybe just to Jason, what are your thoughts on this standard practice for acclimation, background noise, and such?
Yeah, it depends on the particular lab setup. You know, some labs have you know, louder noise air circulation systems than others, or you know, some other distractions that they might want to mask. You don't need it. I mean, if you have a quiet lab, you can use 0 as your background level. If you have a nice quiet, you know, nice quiet lab that you're not going to have any distractions. So I would say it's probably split maybe 70-30 in favor of using some kind of a background level.
Okay.
Well, I'll add a comment there. So the ASR, I mean the San Diego Instruments cabinet has a fan in it. Now, the newer cabinets, they've got a fan in there that's it's quiet, but it isn't 0. We measure it. In fact, I think it's always good to go through your chambers and use the sound level meter to measure whatever your background is with the cabinet fan on. You know, ours hovers around 62 to 65 decibels as our background. Some people will then just decide that they want to set a background level with the programming equipment up to maybe 70. We don't do that, and one of the reasons that we don't is because our lowest pre-pulse signals are just barely above 70, so we don't want to put the background up at 70 for that reason, because then you don't have much difference. So we let the background be as close to whatever that fan you know, produces inside the cabinet as it comes from the company, and we use that as our background level.